Our second group of organic molecules are called lipids. Very diverse group of molecules. They have lots of different functions within the body. They're virtually found in every type of plant and animal cell, performing all kinds of functions. Some examples of that would be we have a certain class of lipids we'll talk about that make up the cell membranes of the plasma membrane around every cell in your body or in every living organism. Lipids are hormones. We're going to talk about lipids as energy storage molecules. There are some that are for protection. And our nerves are actually covered by lipids to insulate them. They serve as an electrical insulation. Very diverse group of organisms, waterproofing and buoyancy. They are hydrophobic molecules, meaning they repel water. They do contain carbon, hydrogen, and oxygen, but we're not talking about the same ratio as carbohydrates. Lipids have less oxygen than carbs. This is an exception. Lipids are an exception to the polymerization that we discussed earlier. Lipids are actually made of two unit molecules. There are two monomers, two groups of molecules that come together to form lipids, but they don't polymerize as did carbohydrates. We don't find very, very huge, large polymers of these. The two monomers come together and form a single molecule. The four groups of lipids that we're going to talk about. First, we're going to talk about fats and oils, what they're made of and what the difference between them are. Then we're going to talk about phospholipids, steroids, and waxes. So let's start by talking about fats and oils. These are lipids that are for long-term storage of energy. Again, we take two smaller unit molecules and they come together and through dehydration synthesis they form a single molecule. We're going to take glycerol, which is this molecule on the left right here. That's a glycerol molecule. And actually three fatty acids bond together with the glycerol to form a single molecule of fat or oil. This is why we sometimes call fats or oils triglycerides. So this is what a molecule of fat or oil would look like right here. Here's my glycerol. Here's my fatty acids, which are basically hydrocarbon chains. You may recognize that. And through the reaction of dehydration synthesis, we've bonded those to the glycerol, and three waters have exited. You may have wondered what is the difference between fats and oils. Well, let's take a look at that. The difference is that one is saturated and one is not. If I were to ask you what a fat was, you would probably say something like lard or butter, Crisco. If I were to ask you what an oil was, you would probably say something like olive oil or canola oil. The primary difference between the two exists because of the fatty acid chains here, and it causes one to be a liquid and one to be a solid. So let's take a look at that. Unsaturated versus saturated fats. Our unsaturated fats come from plants. This would be your olive oil, your canola oil, etc. Notice this fatty acid chain right here. This is what you would typically find in an unsaturated fat. The word unsaturated refers to the fact that these carbon atoms that you see right here are not fully saturated with all the hydrogens they could possibly hold. These two carbon atoms have a double bond between them, meaning that they're sharing two electrons. Well, because these carbons are sharing two electrons with each other, they can only share two other electrons. Remember, carbon only needs 4 more electrons, which causes there to be a hole right here. So this carbon is not saturated with hydrogens. Again, this carbon has a hole also, so those are not saturated. So this is an unsaturated fat. Wherever we see this double bond, it causes the molecule to actually kink where the double bond is. If we look at this space field model of the molecule, this would be our glycerol right here, and these would be our 3 fatty acid chains. Wherever there's a double bond, we actually have a kink in the fatty acid right here. Because these chains are bent, these molecules don't stack so nicely on top of each other when you have lots of them together. These bonds cause this substance to be liquid at room temperature. An unsaturated fat, on the other hand, typically comes from tropical oils and animal fats. We're talking about things like butter and lard. Notice that this fatty acid chain right here would be a chain that would be found in a saturated fat. There are no double bonds between any of the carbon atoms, which means that these carbon atoms are saturated with all the hydrogens they could possibly hold. There are no double bonds, which causes these fatty acid chains to be straight right there. When you have straight fatty acid chains, this molecule would stack very nicely on top of another molecule that looked just like that, kind of like books that you would stack together, which causes these substances to be solid at room temperature. Our next group of lipids are called phospholipids. And whereas our fats and oils were a glycerol and 3 fatty acid, phospholipids are very similar to fats and oils. They actually have a glycerol, 2 fatty acids, and where the 3rd fatty acid would have been, there is now a phosphate group, which is actually a functional group. So it looks something like this. Here's our glycerol, fatty acid, 2 of them, and a phosphate group right there. Okay? Prior to adding the phosphate group, if that had been a molecule of fat or oil, it would have been hydrophobic. But now that we've added this phosphate group, which actually brings a charge with it, the molecule has an area here that is hydrophilic. So when this molecule is made, it forms a structure that is similar to this. A structure that looks like this, where this would be my phosphate group, here's my glycerol, and here's my 2 fatty acids. And what actually has happened here is this phosphate group has bent, coming all the way around, trying to get as far away from the 2 fatty acid chains as possible. So they try to separate from each other because of the nature of the area of the molecule. So we typically call this area of the molecule the head of the molecule. It's hydrophilic. That phosphate is charged, so it likes water. These are the tails of the molecule, and they are hydrophobic. These are fatty acids. They are hydrophobic, so they are repelled by water. Very important that you understand this molecule and what causes it to form this shape, because this molecule, this phospholipid molecule, actually forms the bulk of most of the plasma membrane that covers every cell in your body. In the presence of water, which remember you are 70 to 90% water, in the presence of water, these phospholipids are going to form a double layer. If this is the inside of your cell and this is the outside of your cell and this is actually the covering of the cell, the plasma membrane, this is what the plasma membrane is going to look like. With this being one layer of phospholipids and this being the other layer. So they form a bilayer, we call it. So here's the heads of these phospholipids, and here's the tails, and notice that the heads are all pointing out toward the watery surface because that's the part of the molecule that likes the water. And notice of this layer right here, the heads are all pointing toward the environment, which is primarily water because, again, the heads like the water, the tails hate the water. So with this configuration, the tails are actually insulated from the water, and they never touch the water. I know this is kind of hard to visualize or maybe conceptualize, so let's take a look at it 3-dimensionally. If this is my cell and I took the cell and I cut it in half, inside the cell I would be looking, inside the cell. I would be looking, and all I would see would be the heads of the phospholipids if I had closed the cell. If I closed the cell and looked from the outside, all I would see would be the heads of the phospholipids. So you would never see the tails. They're actually inside the membrane. The only time you would see them would be if you took a cross section of the cell. The next lipid is called a steroid. If you take anatomy, you're going to talk a lot about steroids in anatomy 2, the 202 course. But for today, let's just get an understanding of basically what they are and some examples. A steroid is a lipid that is made up of 4 carbon skeletons. 4 carbon rings right here. So this is estrogen. This is a steroid. Here's testosterone. Here is cholesterol. So those are examples of steroids right there. And notice that this is a carbon ring. This is a carbon ring. Each one of these little points would be a carbon atom. So a steroid is defined as carbon skeletons of 4 fused rings. Our last lipid is called a wax. And most of you know what waxes are or where they're found. Molecularly, a wax is a long-chain fatty acid, which you're familiar with, and a long-chain alcohol. And it would look something like this as far as its structure goes. Waxes are solid at room temperature, and if I were to ask you what are waxes for, or where do we find waxes in your body, you would probably say your ears. And what's it for? It's for protection. It's hydrophobic. It's waterproof. It actually protects your ears. Other places in nature where waxes form, you may have heard of beeswax. The honeycomb is made out of wax. Flowers, plants have wax covering their leaves. Different leaves have different amounts of waxes. Some leaves are waxier than others. If you've ever seen a magnolia leaf or even a camellia, these are very waxy leaves. And again, your ears. Wax is there for protection. It's hydrophobic. It's hard to degrade. It stinks. It doesn't taste good. It deters animals. So it's there to protect your ears.